Hi all, today we are going to start the next chapter, that is reproduction in humans. Compared to the plant reproductive system, human reproduction is also an important chapter because we have to know each and every steps, functions of cells, many things are there. Before entering to this chapter, we have to know why we are studying this human reproduction. We know that reproduction is required, very essential for the survival of the species in this earth, in the face of the earth. Same way we have reproductive assisted diseases, we have reproductive health issues. So if you know come to study about the reproductive system, it is possible to cure some of the problems that exist in humans. We can't say that all of it. We can cure some of the problem that exist in us. We know that very well, and we know that in this modern world reproductive health is becoming a very big major issues. So those who are living in villages, they don't feel much about it, but those who are in the cities, they are facing lot of issues related to the reproductive health, both male and female. So that is the main reason why we are studying this, why we are studying in the biological point of view. Same way, when you are talking about the sustainability of the species on the face of the earth, reproduction is very vital. In India, it will not be a big issue because we are going to become number one in population in another 5 to 6 years. That means we are going to cross China. But when you go to many of the Western countries, the population is very low, very less compared to India. So they are not even meeting the UN standard per square kilometer. That is not maintaining. So they call people from different countries come and settle there, take the benefits from that country. So and many of the reproductive assisted works are also doing nowadays. Western countries have started it, so it is very vital. So in this basis we have going to start this chapter. So keeping one thing in mind, the new syllabus of the CBSE is has been launched. So CBSE is saying not to go in detail much about, not in all topics but in few topics, so there is 30% reduction. So we have to keep that also in mind. We will go into this chapter one by one. So this chapter we can study in two methods. First is taking the male reproductive system, studying it completely. That means what is the structure, what is its function, how the gametes are formed inside the female male reproductive system, then the female reproductive system, then comes fertilization, then the embryogenesis. So like that we can go. That is one way of study. Another way is that first we will take the structural point of view. First we will study the male structure, then we will study the female structure, then in the second level we will study the gametogenesis, both male and female, then fertilization. Afterwards the process goes the same. So here I am going in such a way that first we will study the male reproductive system, what is going inside, what is inside of it, how the sperms are produced. Then we will move to the female reproductive system, fertilization, then embryogenesis, parturition, like that we will go. So we have to study both male and female in detail. So first of all, when we are coming to the male reproductive system, and you know that before entering to those things, we know that we humans are sexually reproducing. That means two sex, we are in two different as male and female, and we are viviparous. Humans are viviparous animals, viviparous organisms. So both the events in male and the female are different according, whenever the sexual characteristics comes, we have a lot of changes takes place in the male and female. And as we know that from time of birth to some age, that is some 14 to 15 age, there will be no much reproductive characters will be there. That means no much of the sexual characters will be there. But after 15, that means from the puberty to the some extent, the human started to show the reproductive change, that means secondary sexual characters, etc., that come to us. So we also have to discuss little bit about that, what which causes secondary sexual characters, importance, etc. We have to study about it. So we will discuss little bit about that. So we will moving first to the male reproductive system. So in the male reproductive system, when we see, there is one diagram is there inside the biology textbook. That diagram is also here. But apart from this, I feel there is one diagram is there which I have copied from the net, which will be more better than that. So it will be colorful, so it will be very easy to understand. So this was taken from the encyclopedia, Britannica encyclopedia. You can see in that encyclopedia also you will get the same diagram, and almost the same explanation what is given in the biology textbook you will get a little more higher level. If you are want to know much in detail in the clinic aspects and all, we have a lot of informations are there, but in plus 2 syllabus we don't want to study that. The clinical aspects little bit only we have to study, not all of it. So if you want to know the clinical aspects, or if you want to make any type of changes inside surgical things or anything we have to do, first we have to know what is male reproductive system, what is exactly going on there. And you know that in the male reproductive system, the major role that is played is by testis. And if you are going to the formation of testis is formed somewhere near to the kidney, and by the time of the birth it will come to the a sac like structure which is known as the scrotal sac. So that scrotal sac you can see here hanging over here inside with the testis. You can see there testis is there, inside that we have the scrotum. So the testis, the male reproductive structure, the testis is hanging outside of the male body. And why it is hanging outside of the male body? Because for the formation of the sperm, the best and the most accurate temperature should be 2 to 3 degree lesser than the normal body temperature. So that is why it is hanging outside of the body. The normal body temperature is recorded as 36.5. If you round off, it is shown as 37. But nowadays it has become very accurate. It should be 36.5. So from that, 2 to 3 degrees Celsius lesser, so that the sperm will be formed very accurately. That is why it is hanging outside of the body, and it is protected inside a sac-like structure known as scrotum. And inside the scrotum we have the testis. So when we go to the testis, when we see the testis from outside, we can see this in the animals. When you go to the butcher shops, etc., when they cut these animals, especially the male, we can see the testis. Whatever we are discussing, we can see it very clearly. So this testis, when the scrotum was cut off and when we go into the testis, we can see that this testis is covered with the white tissue which is known as tunica albuginea. And after that tunica albuginea, when we go inside, we will see the exactly a testis like this, what we are seeing here. And when we go into the testis, we can see testis tubules, that means testis lobules are present, and inside this testis lobules there are fine thread-like structures are called as seminiferous tubules. These seminiferous tubules are the main area where the sperms are produced. In order to produce the sperm, they need few cells inside. One such cell which is very much important for the production of sperm. It is everything is given over here, you can read it. One such cell that is required very well is, it is called, yeah, very much need is Sertoli cells. So these are the cells that give the nutrition to the germ cells. And we have interstitial cells also, which is called as the Leydig cells. So these Leydig cells are responsible for secreting the androgen, that means the male hormone, testosterone. So you have to keep this in mind. In seminiferous, near to the same, inside the seminiferous tubules, lobules, wherever where there is a Leydig cell is there. These Leydig cells are responsible for the production of testosterones. And in the interstitial spaces, that means wherever the sperms are produced inside the seminiferous tubules, we will be having Sertoli cells, which gives nourishment for this growing of the sperm. Uh, so the sperms will be produced in millions inside this seminiferous tubules, and these tubules are connected direct after the formation of the sperm. Somewhere it takes around about 72 hours for producing a complete sperm, one complete sperm. Like that, there will be millions will be produced. And this sperm, now what will happen after producing from the seminiferous tubules, this will be connected, this will be collected, and it will pass us through a network. You can see in the middle there is a network, circular network you can see, which is called it as retitestis. Is clear? Retitestis. From this retitestis, this sperm will be taken to an area known as epididymis. Epididymis, you can see that it is marked over here. It is a coiled region. It is also a networked area, coiled, and it is attached just above the testis. That is why it is known as epididymis. Epi means above, didymis means testis. That is the meaning. And from the retitestis to the epididymis, you can see that tubular structures are going there. These tubular that tubular connection is known as vas efferentia. Vas efferentia. And the sperm, whatever is produced, will be stored inside the epididymis for time being, not for a long time, for a time being. And the sperm will get matured inside the epididymis. Sperm will not be activated, but the sperm will get matured inside this epididymis. After that, you must understand that this sperm has to pass through the penis to the female urethra, not through penis, through urethra to a female vagina. So whenever it is going into the female vagina, the sperm has to get its food. Sperm has to overcome the pH, because the female vagina is very much acidic in nature, so it has to overcome it. It has to maintain the pH, it has to get the nourishment. All these things are required. So whatever the other factors which is responsible for the survival of sperm, it is given by accessory glands. Mainly there are three accessory glands. One is known as the seminal vesicles. You can see the arrow mark what they have shown there. Seminal vesicles, prostate, and bulbourethral gland. Earlier it was known as the Cowper's gland. Cowper's, Cowpers, Cowper's gland. So these glands are very much required. If these glands don't secrete whatever the secretions is required for the survival of the sperm will never function properly. The main function is sperm has to move through the female vagina through the cervix, through the uterus to the fallopian tubule where the fertilization has to take place. So it has to travel a long distance. So whatever it is required, that are given by these accessory glands. So a male reproductive system, we have there is a site for the synthesis production of sperm, and we have the accessory glands are there which will give nourishment to the sperm. So we have to study what are the seminal vesicles, what is its secretion, what they do one by one, then to the urethra, and what is penis and how it is ejaculated out. So this much we have to study. So I think now you understood what is taking place inside the testis. Seminal, we will be having the lobules will be there, tubules will be there. Inside the lobules will be there. Inside the lobules will be have the seminiferous tubules, and the seminiferous tubules are having two cells. Main cells are there. One is the germ layer cells, which is responsible for the production of sperm. Then we have the Sertoli cells, which give nourishment to the sperm. Apart from that, in the interstitial spaces, we will be having the Leydig cells, which are responsible for the which secretes androgen. So we will discuss about that, how this androgen is secreted and what are the hormones responsible for the production of this sperms. So now we will come to the seminal vesicles, which is an first accessory gland. So when we see the seminal vesicles, these seminal vesicles secretes mucus and watery alkaline fluid which contains nutrients including the sugar fructose, which is the energy source, and some other chemicals which are helping for the movement of the sperm inside the vagina, female vagina. So if you look very detailed, we can see that the seminiferous tubules are secreting these. One is fructose, one is alkaline fluid, then a protein like seminoglycan, which gives a sperm a jelly like so that it can stick somewhere. Then phosphorus and potassium, they are minerals. Then prostaglandin, which plays a role for the movement of the sperm inside the vagina. Prostaglandin, may play a major role in lowering the response of the female, that means which helps to the movement of sperm inside the vagina. So these are the main thing what is secreted by the seminal vesicles. Now when we are coming, and one more thing, fructose sugar will be seen in a male body only in one area. Through through the digestive system, when we take fruits, we will get fructose, but it will get digested and absorbed. But outside of the body, we can see fructose only in one area, that is inside the semen. So whenever it is a clinical part, so whenever what we call a rape incident or something is taken place, so they will check whether fructose is present inside the female body, that means in vagina. So if the fructose content, if they can find out, they can say that sexual intercourse has happened. So whether it is a rape or other, they can't confirm, but they can say that the sexual intercourse has happened. So fructose they will check for the first time. Then when we are coming to the prostate gland, this prostate gland secretes the alkaline fluid, almost the 70% of the volume, whatever the required for the semen will be from the secretion from the prostate, which secretes an alkaline solution, and it gives lubrication to the sperm and help to neutralize the acidic and the vaginal content. That means whenever alkaline and acidic both will come together, it will become a basic ending. Next comes is the bulbourethral gland. This bulbourethral gland, you can see the position in the this diagram, which is very near to the beginning of urethra. That means it is structured somewhere near to the beginning of the penis, where the muscles of the penis are coming out. So what is the main function of this bulbourethral gland is, whenever the sexual intercourse is taking place, these bulbourethral glands secrete some glycoproteins which will help to give lubrication to the penis. And another thing is that when the bulbourethral gland is secreting these glycoproteins, it also expels out if there is any dead cells are there in the urethral tube, so that the sperm can come out very properly. So these and this secretion will be this secretion will come as soon as the penis get aroused for a sexual intercourse. So it will happen very before the sperm will ejaculate, before the ejaculation of the sperm. And all these secretions of the three glands, we will together call it as seminal plasma. And after all the secretion of this, the pH of the sperm will be alkaline, somewhere between 7 to 7.1 to 8. 7.1 to 8. So this makes the sperm, it gives strength to the sperm. Now the sperm is having everything. So the next the thing that through which the sperm will be ejaculated out, that is the penis. So when we see this male penis, these male penis are made up of erectile tissue, that is spongy tissues. Spongy tissues means these spongy tissues are having gaps inside. So whenever the blood runs inside these spongy tissues, these sponge tissues become very hard. So penis will become very hard during sexual intercourse in any animals, in humans also, even in other organisms also. And the if you see the penis, the tip of the penis, enlarged tip of the penis is known as the glans penis, and it is a loosely fold with a skin known as foreskin or purpose. So there are some traditions are there inside the Muslims and even in the Jewish system also. They will cut this foreskin. It is according to the religious traditions they are doing that. So it is practiced. So these are the main content of the male reproductive system and its accessory glands. I think you have understood it. Next thing after this we are going to discuss is spermatogenesis. Spermatogenesis means how the sperms are formed, then the structure of the sperm. With this we will conclude. So before entering to the spermatogenesis, we have to know about the hormones, which we have studied in +1. So whatever the changes has to takes place inside a male or a female reproductive system, all the major roles are played by hormones. Without hormones, nothing. So I hope you can recollect something from the +1. You have studied about GnRH, gonadotropin releasing hormones. So GnRH will be secreted from the hypothalamus, and it will pass through anterior part. That means it will be stored inside the pituitary gland, that is anterior portion of the pituitary gland. So when it reaches, it is not stored, it will signal the anterior part of the pituitary gland. From this anterior part of the pituitary gland, and another hormone will be secreted which is known as gonadotropins. So there are two types of gonadotropins are there. One is follicle stimulating hormone which is known as FSH, and the luteinizing hormone which is known as LH. So for in both male and female these hormones are same, they will do some functions. For example, when the follicle stimulating hormones are secreted, FSH is secreted, these serotonin cells, they started to mature. They will give the signal in the one of the process in spermatogenesis to make matured sperm. So whenever the LH is stimulated, it will go and stimulate the Leydig cells to secrete testosterones. Testosterones. So and one more thing you have to understand that in both male and female we are having both testosterone and estrogen. There is a percentage of estrogen is present in the male also. There is a percentage of testosterone is present in female also, but the amount of testosterone will be more when we compare to the estrogen. So there are a lot of medical side things are also there related to that increase and decrease of this. So just to know about this you have to keep, because in the lower class textbooks you have studied that the male hormone is testosterone and the female hormone is estrogen. It is on the basis of the content, the amount, how much it is. So both are there, and you might have studied about that. If the testosterones are not secreted properly in both male and female, sexual libido, that means sexual pleasure will not be there. I think this is given in the last portion. Neural control, hormone coordination, control and coordination plus one, it is given some few lines in CBSE is given. And if the testosterone and estrogen level is not accurate, the spermatogenesis also will not takes place properly. So these hormones play a major role. So these hormones comes from the master gland, that is pituitary. Follicle stimulating hormone will go and stimulate the serotonin cells, and the luteinizing hormone will go and stimulate the Leydig cells. So one will started to produce testosterone. When testosterone is started to produce, the male secondary sexual characters started to come. So when the testosterones are started, then the serotonin cells will start to mature to form, help nourish the cell to form more and more matured sperm. So this is the concept. The same way there is a feedback system is also there. For example, if the amount of testosterone secreted is more, so for everything there is a level, we can't cross that level. So whenever the amount of testosterone increases, the serotonin cells will secrete a hormone which is known as inhibin. It will go and signal to the pituitary gland to reduce the secretion of FSH and LH. So that particular hormone is known as inhibin. Serotonin cells will secrete. So when it is secreted, it will, and sometimes even the testosterone also will give a negative feedback to the pituitary. So it will not form much major issues, but when inhibin is secreted, it will go to the pituitary gland and it will make the reduction of FSH. So FSH is the main hormone which is nourishes the sperm, nourishes spermatid to form into spermatozoa, that we will study in spermatogenesis. So by keeping this in mind, we are moving to the spermatogenesis. So if this is the diagram of spermatogenesis, and sidewise you can see the transverse diagram, what is exactly taking place inside that, which is the copy of the first diagram where we have discussed. So spermatogenesis, as per the examination point of view, each and every point is very very important, each and every step what is happening. So as we know that when the child is born, child is born, there will be few cells will be there which is responsible to grow into a sperm. So from that, from where this sperm, that means we can call it, we can call very directly it as a germ cell. Germ cell means which have the capacity to differentiate into one particular tissue in sorry one particular tissue or an organ inside a body. So which is responsible for synthesizing the sperm, that means which cell which have the capacity to differentiate into a sperm, that particular cell is known as spermatogonia. And spermatogonia will be 2n in condition, that means it has not even undergone meiosis. And this 2n in condition, again they will divide into primary spermatocyte, which is also 2n in condition. Why they are undergoing these type of division? Because even in the male, if you see inside the male system, we can see that the till the puberty, and even in the male, old age also doesn't matter to produce spermatozoa, that means a matured sperm. So till the lifelong it has to go. So they need more cells, more cells and tissues which have to form into a sperm. So they will go like with a backup plan. So if the 2n is there, that 2n will again divide it into spermatocyte, and these 2n have the capacity to form a spermatogonium bag, or otherwise that spermatocyte will form to a matured sperm. And this spermatocyte will undergo meiosis 1, where the crossing over is taking place. It will form into a secondary spermatocyte, and this secondary spermatocyte will undergo meiosis 2. From the one, two spermatids will be formed, and this spermatid will undergo spermiogenesis, form spermatozoa. So when that hormone, with, before that we have discussed about that, that is this follicle stimulating hormone Sertoli cells. These Sertoli cells will come in action during spermatid, that means in spermiogenesis, from where the matured sperm is formed. So there this Sertoli cells come into action. They will nourish the spermatids, they will give energy to form a matured sperm, which is called a spermatozoa, through the process of spermiogenesis. And this spermatozoa will travel through the seminiferous tubules to retitestis to epididymis. It will store there for a time being. After that, it will, whenever the sexual erection comes, that will vas pass pass through this secondary accessory glands through vas deferens is the tube. I think I forgot to say that. Vas deferens is the tube structure that carries the sperm from epidermis through the urethra. That means whenever the sperm is passing through that, the accessory glands will secrete whatever the secretions are required. Is it clear? So this thing you have to study very properly. Mitosis, meiosis, where it is taking place, what is my meiosis 1, what is meiosis 2, this thing you have studied in +1. Then we'll coming to the structure of sperm. This you have studied, you know that. If you see the sperm will be having two parts. One is head, three parts. One is head, middle portion, and a tail. Tail is for the movement, which is having 9+2 arrangement. So if you see in the head will be having a nucleus. A nucleus will be there, which is having the 23 pairs of chromosome. Ploidy is 23 after meiosis. and it will be has a plasma membrane, and it is also covered with a structure on the head which is called as acrosome. This acrosome will be fit to the head of the sperm inside the epididymis. If the acrosome is absent, we can't say that the sperm is a matured sperm, because if the acrosome is absent, the sperm can go and hit and penetrate the ovum inside the female body. So they secrete some enzymes which you known as hyaluronase enzymes, which have the capacity to break the outer layer of ovum and penetrate in. And after that, we will be having a centrioles. We know that what is the function of centrioles, from where the spindle fibers arises. Then after that, we'll be having mitochondria. Why the mitochondria is required? Mitochondria is required to produce the energy. For the energy, what is the sugar source? That is fructose, which is secreted by the seminal vesicles. Then we'll be having the flagella. Flagella, why it is flagella is required? For the movement of the sperm from the vagina through the cervix, uterus, to the fallopian tubules till it reaches to the ovum. So this is the main structure of sperm. And the first person to see the structure of sperm under the microscope, the person is Antony van Leeuwenhoek, the same person who has seen the first protozoan or first living organism under the microscope. The same person has seen this. So this is the main part. And one more thing, I said that in some condition, if you see in some organisms, like if you see majority of the organisms like humans, bull, dogs, etc., the testis is holded outside of the body, which is known as scrotum. In some organism, this testis will never come out. That means it will be inside the body. That means there will be no scrotum. It will not be hanging outside. Such condition is known as testicont. So like whale, dolphin, etc., we can't see any testicles outside. That will be inside of the body, which condition is called as testicont. Another one clinical condition is there. I have said that the testis will be formed near to the kidney, and it will be pushed to the scrotum by the end of, by almost the, when the baby reaches to 8 to 9 months, or within, after birth, even within 1 or 2 months, that testis will be pushed into the scrotum. In some conditions, the testicles will not be pushed into the scrotum. That is undescended testicles. So that particular condition is known as cryptorchidism. Cryptorchidism. So these things you have to keep in mind. If there is any doubt or any topic I have made mistake, or if you don't understood anything, please comment wherever I am, below wherever I am posting the video. Thank you. And one thing is, please read textbook very carefully. You will get maximum questions from the textbook only for the biology point of view. So please read. Any doubts, comment me back. Thank you.